Hello, this is lecture 10. This is the last lecture in this unit or section on regulation of gene expression. Up to this point, we've talked about regulation of gene expression by looking at regulation at the transcriptional level. Now we're going to look at regulation at the post-transcriptional level. This slide here shows that we're going to encounter microRNA, which can bind with mRNA and affect whether or not that mRNA is going to be used to make a protein. And we'll see how that works a little bit later on. So if you take a look at this slide right here, everything in the nucleus we've talked about so far. We've talked about initiation of transcription. We've talked about processing of the mRNA. We've the capping, the elongation, the splicing. We've talked about polyadenylation and how that's associated with termination. And so when all is said and done, what you end up with is this mature mRNA. And this mature mRNA has the cap on it, has the cap on it, has a poly A tail on it, and the introns have been removed. So that's a fully processed, mature functional mRNA. That mRNA is going to get exported from the nucleus where, as we know, it's going to be translated into a protein. But the question is, once that mRNA is exported, is that the end of the ability of the cell to regulate expression of that protein? And if you think about it, it would seem that maybe there would be other ways to regulate how much of that protein the cell is going to make. In other words, the cell usually has backup, or you might call them redundant systems, for ensuring that it can tightly regulate a process that needs to be regulated, in this case, how much of a protein we have. And so the answer is, really, once you make that mRNA, let's just put our mRNA here. We'll go ahead and put the poly A tail on it. We'll do this to designate the cap. We know that's going to be translated into a protein. But there are ways that the cell can actually control how much protein we make, even after that mRNA has been made. And these are really going to fall into two broad categories. One has to do with stability of the mRNA. And just to summarize, if it's not stable, that means it's going to be degraded. And of course, if it's degraded, you can't translate it to make a protein. So controlling or regulating mRNA stability is going to play an important role in this post-transcriptional regulation. And the other way would just be controlling translation. In other words, if you've got an RNA, even if that mRNA is stable and is not going to be degraded, you're still not going to get a protein unless you translate that mRNA. So in just using broad strokes here, we can appreciate the fact that this is what the cell is going to do. We're going to make that mRNA, and then that mRNA is going to be either stable and hang around for a while, in which case it could be translated into a protein, or it's not going to be stable and it's going to be degraded. And if it does hang around for a while, the cell can even control whether or not it's translated. So that's what we're going to deal with in the next few slides. Now, before we look at those mechanisms, let's just think about this mRNA and what an export-ready mRNA would look like. This really is kind of a review, but it also sets the stage for the process of translation. So let's begin over here on the left. And look at this protein. What's going to be the indicator that this protein is actually ready to be exported? Well, there are several things. One is that we've got the 5' cap that we talked about, but there is also a protein called the cap binding complex. So the cap binding complex is going to bind to that 5' cap. we can see so the cap is there, obviously. In order for the cap binding complex to bind, we've got our poly A tail right here, and we've got our poly A binding proteins. We're going to need a nuclear export receptor to get that out, so that nuclear export receptor is going to bind. And then you'll notice on this slide we've got some SR proteins and the exon junction complexes. We talked a little bit about those the other day. So in order for this protein to be export ready, as it's being exported, some of these things that are no longer needed are going to be removed. So for example, the SR proteins are no longer needed. They're going to be removed. The exon junction complexes are going to remain. The nuclear export receptor, of course, is going to be used to actually get the protein out. And as the protein comes out, some of these, as the mRNA rather comes out, some of these proteins are going to be dislodged. Okay, so in essence, you can summarize this mRNA this way. An export-ready mRNA. It's going to have our poly A binding proteins. Those are not only important, as we'll see a little bit later on, but they also are an indication that the poly A tail has been added. It has a cap binding complex, which is important for export, but also indicates that the 5' cap is there. It has exon junction complexes, and those exon junction complexes, remember, are going to remain on the mRNA where two exons were joined together. So that's an indication that splicing has occurred. And then there should be an absence of these snRNPs, these small nuclear ribonucleoprotein particles, because we know they were involved in splicing. Well, if splicing is complete, then they should be gone. And so essentially what's going to happen is, aside from the proteins that are involved in export, that's kind of what your export-ready mRNA should look like. Now, when that export-ready mRNA gets out into the cytosol, almost immediately what's going to happen is the cap binding complex is going to be replaced by a couple of really important proteins that are going to play a role in translation. So this is EIF4E and EIF4G. Okay, and notice how EIF4E is attaching, is essentially replacing the cap binding complex and binding to the 5' cap. Notice that EF4G is bound to EIF4E, and then EIF4G is going to bound to these poly A binding proteins, as you can see right here. So we're going to end up with what's called it's a circular mRNA. And that circular mRNA is then ready for translation. Now we'll talk a little bit later about the circular part of this. But the point in this slide is just simple. Once the mRNA is fully processed and ready to go, some proteins are going to be involved in exporting it. Some of those proteins are going to get kicked off as it moves through the nuclear pore complex, which we'll talk about later. And then what's going to happen is the cap binding complex is going to be replaced by these two other proteins that are going to create this circular mRNA. Now, we're not going to spend a lot of time talking about translation. You get that in other courses, but we're really interested in here in regulation of translation, or specifically post-transcriptional regulation. So we've got to spend a little bit of time talking about the beginning of translation. So this is a complicated looking slide, but it's really not as bad as it looks. So let's just kind of begin right here. This would be the mRNA that we saw on the previous slide. In other words, notice that it has your poly A tail, it has your poly A binding proteins, and it has EIF4E and EIF4G bound to the 5' cap, and then creating the circular structure as they interact with the poly A binding protein. That's what we saw on the last slide. Now, for the sake of discussion here, we're going to pretend that is not circular, that it's linear, just because this is going to make it a whole lot easier. Now, what's going to happen is the small ribosomal subunit is going to bind to the 5' end. And that small ribosomal subunit is going to bring with it the initiator methionine, and it's also going to bring with it a protein called EIF2, which is a G protein. We've alre' talked about G proteins. So this EIF2 will become important a little bit later on. Okay, so that's going to assemble at the 5' end of this mRNA. Now the next thing that happens is that small ribosomal subunit that contains that initiator tRNA is going to start moving toward the 3' end. It's scanning for the start site of translation. Now we drew this on the board a couple times, but remember, this is the 5' end. This is the 3' end. Translation is going to begin somewhere downstream of the 5' end. So this small ribosomal subunit is moving in this direction looking for the start site. Now the question is, what is it looking for? Well, it's looking for what's called the Kozak sequence. So let's think about what the Kozak sequence is. It's usually within about 100 nucleotides of that cap. It's usually the first AUG downstream of the 5' end. That's true in about 90% of eukaryotic mRNA. Now what that means is it doesn't have to be the first AUG. It could be the second AUG. It's going to be that AUG that it's looking for is going to be preceded by an ACC and followed by a G. Okay, and the bold A right here and the bold G right here really help to identify that as the starting AUG. So this whole sequence, including that bold A and that bold G, make up what's called the Kozak sequence. It was discovered by a scientist named Marilyn Kozak, and that's where it gets its name. And so that's what it's doing. It's scanning, looking for the Kozak sequence. Now, if the sequence differs from this substantially, you can get what's called leaky scanning. You can get two different proteins with different amino termini from the same mRNA. In other words, if this was, let's say this is the Kozak sequence right here. Well, let's say this is an AUG and this is an AUG. But let's say that the sequence surrounding that first AUG kind of looks like a Kozak sequence, but not exactly. If the translation machinery can't tell, then it may use that as the starting point for translation, or it may pass over it and use the second one. And if that's the case, we're going to get a protein that looks like this or a protein that looks like this. So you can have two different proteins in the cell with different amino termini, depending on whether or not the translation machinery correctly identified an AUG as the first AUG to be translated or not. In other words, did it correctly identify it as the start site, or did it skip over it and use the next AUG? Now, a little bit later on, we're going to talk about something called an internal ribosomal entry site. Now, that internal ribosomal entry site means that the ribosome is not starting at the 5' end and scanning for the AUG. It can skip that. Now, that's not going to be relevant when it comes to post-translational or post-transcriptional regulation of gene expression, but it is going to be relevant later this semester. So just remember that most of the time, or for most mRNAs rather, what we can say is that translation occurs when the small ribosomal subunit binds to the 5' end, scans looking for the Kozak sequence. Once it gets to the Kozak sequence, then what's going to happen is the small ribosomal, the large ribosomal subunit is going to then be added, and this EIF2 is going to be removed. Remember, this is a G protein, and when the GTP, when the small ribosomal subunit and the EIF2 reach the Kozak sequence, the GTP gets converted to GDP. That G protein becomes inactive, dissociates from that initiator tRNA, and that allows the large ribosomal subunit to be added. Now in other classes you can talk about this whole process of translation. We're not going to talk about that, how different amino acids are added. I know that's covered in genetics and some other courses, but we really had to talk about what happens here at the beginning, because this is going to play an important role in post-transcriptional regulation of gene expression. Now, before we go on, let's take a look at this slide, which just kind of shows you that the mRNA really exists not in a linear form, but in this circular form. And so the sequence of events that we just described are going to start right here on this end, and the scanning is going to be moving in this direction, and you can see there's our start codon. Okay, and as this ribosome begins to translate, it's going to go all the way around. It's going to get back over here, and you can see that the further it's gone, the longer the protein is. This is the protein right here. Okay, now the beauty of that is once you get to the stop codon and the ribosome disassembles, it's actually, everything now is back in place where it needs to be to start again. So it can just hop back on and we can start again. The other thing to notice from this is that once a ribosome moves out of the way, that allows another ribosome to come on. And so what you've got here is an mRNA that's being simultaneously translated by multiple ribosomes. And so this is called a polysome, poly meaning many, or polyribosome. Okay? You can actually see one of these over here. Now that we know a little bit about the way translation begins, we've got to go back now and think about this concept called the steady state level of the mRNA. Now, steady state is just a general term that means the concentration of something doesn't change. It's constant. The concentration doesn't change. The analogy that I like to use every time I do this is a bathtub. So think about a bathtub that's halfway full of water. If we turn on the faucet and cause more water to come in, then there's going to be more water in the bathtub. So that's not a steady state. If we open the drain and allow water to drain out of the bathtub, then the amount of water in the bathtub decreases. That's not a steady state either. But if no water is coming in and no water is leaving, then we've got a steady state. Or the other way to have a steady state is to have the amount of water that comes in through the faucet be exactly equivalent to the amount of water that leaves the bathtub through the drain. In that case, you're still going to have a steady state. In other words, you have input into the system, but the amount of input is balanced exactly by the output. And so the steady state remains. So the steady state label or level of mRNA is going to be determined by the rate of transcription. That would be analogous to water flowing into the bathtub. And the rate of degradation. That would be analogous to water going down the drain. So in other words, how much mRNA exists in the cell is a function of two things. The rate at which the mRNA is made, and of course that's going to be regulated by transcription, and the rate at which the mRNA is degraded. So there's a concept called mRNA stability, and mRNA stability plays a really important role in determining the steady state. So a stable mRNA is an mRNA that hangs around for a long time. It doesn't get degraded rapidly. So so we can talk about half-life. If an mRNA is stable, then that means it has a long half-life. It means its degradation is slow. Okay? So in other words, let's say we got this mRNA right here. Let's say it's a really stable mRNA. If it's a stable mRNA, it means it's not going to be degraded, which means it has a long half-life. Now let's think about that term half-life. Half-life is the length of time it takes for half of something to disappear. We normally think about this when it comes to isotopes. So if you have a radioisotope, then the length of time it takes for half of that isotope to decay is called the half-life. The analogy that I normally use in class is, let's say we've got a class and it's got 60 people in it, a classroom with 60 people in it. And let's say the bell goes off, and when the bell goes off, the people leave the classroom. Well, the length of time it takes for half of those people to leave the classroom would be referred to as the half-life. Okay? So if you think about something with a long half-life, then that means it's going to be degraded slowly. It means that if it's an mRNA, it means it's going to hang around in the cytoplasm for a long time. Now that's important to think about, because that means that if that mRNA is stable and hangs around in the cytoplasm for a long time, you could actually be translating that mRNA and making protein way long after transcription has ended. In other words, transcription has occurred. We know how that regulation occurs. Maybe that has stopped. Maybe the gene is no longer being transcribed. But if that mRNA has a long half-life, that mRNA could still be in the cytoplasm and being translated for a long time. Now let's see what happens if you've got an mRNA with a short half-life. This means it's going to undergo rapid degradation. It means it's going to be unstable. Now that can result in rapid changes in protein concentration. Because let's say you've got an mRNA, and that mRNA is being translated into a protein, but if the mRNA is rapidly degraded, then obviously it can't be translated, and so you're not going to be able to make that protein. And so if you wanted to have a constant supply of a protein that was derived from an mRNA with a short half-life, what would you have to do? You would have to constantly be transcribing the gene to make more of the mRNA. So you can see the rate of transcription of the gene and the stability of the mRNA are the two things that are going to control how much of that protein you have. Now there's one other thing a little bit later on. That's going to come to whether or not that mRNA is translated. But for now, when we're thinking about stability and transcription, we can see how those two things work together to determine the concentration of mRNA. Okay, so a typical half-life for an mRNA is about 30 minutes. Now, we'll come back to this idea of half-life in just a few minutes, but before we do this, let's think about degradation of RNA. How would an RNA be degraded? Well, we know that the cap is going to protect the 5' end. We know the poly A tail is going to protect the 3' end. Now, here's what's really important. It's going to come up over and over. As an mRNA gets translated, each time it gets translated, the poly A tail gets shorter and shorter and shorter. This is why it says gradual poly A shortening. Now imagine what's going to happen here. As the poly A tail gets shorter and shorter, it's going to lose its ability to protect the 3' end. Now, as we'll also see a little bit later on, remember, it's not this mRNA doesn't exist in a linear fashion like this. It's really a circle with that poly A tail interacting with the 5' cap. Remember EIF 4E and G. So as that poly A tail gets shorter, that interaction is also going to be reduced. That's going to be important as well. But let's focus on what happens here specifically with the poly A tail getting shorter. As the poly A tail gets shorter and shorter, once it gets down to about 30 A's, then it really kind of loses its ability to protect the end. And so what happens in that case is you can get what's called 3' to 5' degradation. 3' to 5' means that we're moving in this direction. So nucleases that are going to degrade the mRNA are going to start chewing it up from the 3' end. This is the 5' end, this is the 3' end, right? So they're going to start moving in this direction. That's 3' to 5' degradation. Now, if an mRNA is decapped, then we can get degradation in the other end as well, right? And as we just saw, and as I just said, we haven't really seen it yet, but remember that circular RNA with the 5' cap that has EIF4E and EIF4G attached to it. We said that that's going to attach to the poly A tail. As the poly A tail gets shorter, then that's going to eventually lead to decapping, because there is no poly A tail to interact with the 5' cap anymore. And without that interaction, the 5' cap is going to be removed, and now we can get degradation in that direction. This is from a different textbook, but I really like this because it elaborates and shows this a little bit more. So let's start out here in the middle. This is what's called deadenylation, a deadenylation dependent pathway. This is exactly what we said. The poly A tail gets shorter every time it gets translated. And because it gets shorter, we can have 3' to 5' degradation. Now this is called exonucleolytic decay, and it's called exonucleolytic decay, exo meaning coming from the outside. So this is coming from the outside, getting exonucleolytic decay. The enzymes are actually part of a structure called the exosome. We're not going to get into detail on that. Suffice it to say that these nucleases that are part of the exosome will cause this degradation to occur in the 3' to 5' direction. Now we just said you're going to get decapping as your poly A tail gets shorter. And as your poly A tail gets shorter, we're going to get degradation in this direction. So this middle section here of this slide is exactly what we saw on the previous slide. It's called deadenylation dependent degradation, because both ends, degradation on both ends occurs because you've lost the poly A tail. Now let's go over here. This is deadenylation independent degradation. In this case, anything that causes the 5' cap to be removed is going to allow it to be degraded in the 5' to 3' direction. In other words, sometimes the cap can be removed independently of the poly A tail. And so that's why this is called deadenylation independent. In other words, decapping that occurs without any changes to the poly A tail are still going to result in degradation. Again, this is 5' to 3' exonucleolytic decay. It's going to happen here, and it's going to happen here. Now what about on the other side? This is endonucleolytic. If exonucleolytic decay occurs from the outside or from the ends, that's what we mean by that, from the ends, then the endonucleolytic pathway occurs whenever some protease, not protease, but nuclease clips the RNA. And if it clips the RNA, as you can see right here, then the ends are no longer protected. This end right here would no longer be protected, and we can get degradation in the 3' to 5' direction. So this is an endonucleolytic pathway. So here's the point in all this. There are nucleases that will destroy these mRNAs. And whether they're able to destroy these mRNAs really depends on whether or not we can maintain the cap and the poly A tail. If there's anything that causes the poly A tail or the cap to be released, then we're going to get nucleolytic decay. Now, again, let's go back and look at this. Here's our EIF4E and our EIF4G. We've already talked about those. We're going to get this circular RNA, and so we could get translation here. Okay, this slide is just attempting to show that there's essentially one of two things is going to happen. You're either going to get that mRNA translated, or it's going to undergo decay. So again, what's happening over here on the right is that the poly A tail, because Excuse me. In this case, the poly A tail is being destroyed by a deadenylase. Okay, so notice what's going on here is the poly A tail is being destroyed. And if that poly A tail is destroyed, then as we said, you can get 3' to 5' exonucleolytic decay, and the cap is then going to be removed, and you'll get 5' to 3' decay. Now, many short-lived mRNAs, so these would be mRNAs with short half-lives, have sequences in the 3' UTR that bind proteins that interact with these deadenylating enzymes and the exosome to promote degradation. Okay, so in other words, here's your mRNA. This is your 3' UTR. 3' in. This is where translation would stop right there. But if this 3' UTR has this AUUUA sequence in it, that's going to serve as a binding site for proteins that induce the deadenylation. And that induction of deadenylation then is going to lead excuse me, to degradation, as we saw on the previous slide. Now, to bring all this together and see how this works, we're going to look at a classic and well understood example. This example involves iron. So it turns out that iron is required for cells to function. And so if a cell has an iron deficit, it needs a mechanism for bringing iron into the cell. But the problem is that iron can also be toxic. So if a cell has too much iron, then the cell has to have a way to deal with it. So there are two really important proteins here. One is called the transferrin receptor. Okay. The transferrin receptor binds transferrin and brings iron into the cell from the circulation. So in other words, the transferrin receptor binds transferrin, and transferrin exists in the circulation bound to iron. Okay? So let's imagine we've got a cell down here. There's the nucleus. On the surface of that cell, we have a transferrin receptor. Okay? We'll say that transferrin is yellow, and we'll say that iron is green here. So there's the transferrin receptor binding to transferrin, and that transferrin is going to bring an iron molecule in with it, iron atom in with it. Okay? So what's going to happen then is this iron and this transferrin are going to get into the cell. They'll dissociate, and then that iron can be used by the cell. So if you don't have enough iron in your cells, then what's going to happen is you want to upregulate the number of transferrin receptors so you can bring more iron in. Now, we said iron is toxic. So it turns out too much iron is toxic. So it turns out that if we've got too much iron, we need to bind that iron. Okay, so we'll just use this purple thing here. And we'll call this, I shouldn't use purple because let's do this differently. Okay, here's iron. Let's use this light blue thing here. This light blue protein is ferritin. Ferritin is an iron storage protein. So ferritin is going to bind iron, sequester iron, and it keeps iron from damaging the cell. So if you've got too much iron, then you want to upregulate the amount of ferritin that you have. But you can see that the cell is going to have to strike a balance between upregulating the transferrin receptor and upregulating ferritin. So the cell has to engage in what's called coordinate regulation of gene expression. In other words, the cell has to have a way to coordinately regulate how much transferrin receptor and how much ferritin you have. Because another way to think about this would be if you have an iron deficit in the cell, not only do you need to upregulate the transferrin receptor to bring more iron in, but you need to downregulate ferritin, because you don't want that ferritin to bind to the iron if you already have an iron problem. Conversely, if you have too much iron in the cell, you need to upregulate ferritin so that you can bind to that iron to keep it from being toxic. But at the same time, you want to downregulate the transferrin receptor so that you're not bringing more iron into the cell. So the cell has to coordinately regulate these two proteins. And the way it does it is absolutely fascinating. So let's first start out, by looking at an iron starvation condition. In this iron starvation condition, the cell doesn't have enough iron. So if it doesn't have enough iron, what does it need to do? We want to downregulate ferritin, upregulate the transferrin receptor. So it turns out that there is a protein called cytosolic aconitase. Here is cytosolic aconitase. Now, notice that cytosolic aconitase can bind to the 5' UTR of the ferritin mRNA. So here's the ferritin mRNA over here. And cytosolic aconitase is binding to the 5' UTR. Cytosolic aconitase can bind to the 3' UTR of the transferrin receptor mRNA. Now remember, we're under iron starvation conditions, which means we don't have enough iron. So we don't want any ferritin protein under these conditions. So it turns out that when cytosolic aconitase binds to the 5' UTR, we block translation of the ferritin mRNA. Now, how would we do that? Well, remember what's going to happen. We're going to assemble on the 5' end of the mRNA the small ribosomal subunit. Remember, that small ribosomal subunit is going to start scanning, looking for the Kozak sequence. Well, the Kozak sequence is going to be right here, right? Because that's the start site of translation. Well, it turns out that when cytosolic aconitase is bound to the 5' UTR, it serves as a roadblock. In other words, the small ribosomal subunit cannot continue to scan past the cytosolic aconitase. And so it never gets to the Kozak sequence, so it never starts translating the mRNA. So this is a block in translation, and no ferritin is made. Now on the other side of the coin, you need to upregulate the transferrin receptor. Well, here's what's going to happen. The cytosolic aconitase binds to the 3' UTR of the transferrin receptor mRNA. Now, the transferrin receptor mRNA is one of those mRNAs where there's an AU-rich sequence in the 3' UTR. Now remember what that AU-rich sequence does. That AU-rich sequence in the 3' UTR serves as a binding site for deadenylating enzymes and binding of the exosome, which leads to degradation of the protein, of the mRNA. So it turns out when cytosolic aconitase is bound to the 3' UTR, it covers up those AU sequences, and consequently, neither the exosome nor the deadenylase can bind. So what does that do? That preserves the mRNA. It keeps it from being degraded. Said another way, it increases the half-life of the transferrin receptor mRNA. And the longer it hangs around, the more it can be translated. Okay, and it can be translated just fine because cytosolic aconitase doesn't bind to the 5' UTR here. So what's going to happen here? Under iron starvation conditions, cytosolic aconitase is blocking translation of the ferritin mRNA, and it's inhibiting degradation of the transferrin receptor mRNA. Or said another way, it's increasing the half-life of the transferrin receptor mRNA. Now, what about conditions in which there's too much iron? an excess of iron. Well, here's what happens. In the excess of iron, the iron will bind to cytosolic aconitase. In other words, cytosolic aconitase has an iron binding site. Now that wasn't an issue up here. Here's the iron binding site right here. Well, let me do that differently. Here's the iron binding site right here. But notice there's no iron bound to it, and there's no iron bound to it because you're under iron starvation conditions. But if you're under a condition in which you have too much iron, then the iron binds to that iron binding site. And when it does that, it causes the cytosolic aconitase to have a reduced affinity for both the 5' UTR of the ferritin mRNA and the 3' UTR of the transferrin receptor mRNA. What's going to happen now? Well, with cytosolic aconitase gone, the ribosome can scan looking for the Kozak sequence. It'll find the Kozak sequence. It'll translate that mRNA and make more ferritin. In the case of the transferrin receptor mRNA, with the cytosolic aconitase gone, the deadenylating enzymes can bind. They're going to deadenylate the transferrin receptor mRNA. In other words, we're going to reduce the length of the poly A tail. And what's going to happen then in that case is that that mRNA is going to be degraded, so no transferrin receptor can be made. No transferrin receptor can be made because the mRNA has been degraded. It's not there anymore, so there's no mRNA to be translated. So this is a beautiful and well understood example of coordinate regulation in which half of the equation involves controlling translation of an mRNA, and the other half of the equation involves controlling the degradation of another mRNA. Okay, so on the last slide we saw that the transferrin receptor mRNA gets degraded. So anytime there's degradation of mRNA for whatever reason, either because it's been decapped or because the poly A tail has been shortened, which eventually leads to decapping, for whatever reason, it turns out that that degradation of that mRNA doesn't take place just randomly in the cytosol. Rather, it takes place in specific structures known as Pbodies. Pbodies, I've described Pbodies here, are large assemblies of mRNAs and mRNA degrading enzymes. Now that's a recipe for destruction, right? Because you have the substrate that needs to be degraded, the mRNA, plus the enzymes that do the degrading. And so these compartments within the cell, they're not organelles because they're not membrane bound, but they're localized compartments. Kind of like we saw the nuclear neighborhoods in the nucleus, where things are just assembled and held together, usually with some sort of scaffold protein. Well, you got the same sort of thing going on here. In this slide, there are decapping enzymes those proteins involved in decapping are stained with an antibody and it shows up green. In this one, the same cells are stained with an antibody to a protein called argonaute. Now, we're going to see a little bit later on, we haven't seen this yet, but argonaute is also involved in mRNA degradation. We haven't seen it yet. We'll see it in a couple of slides from now. But the point is, argonaute, which is involved in degradation, and the decapping enzymes end up co-localizing in cells. So we can see that wherever you see the green, like these three, they show up as yellow here if you superimpose these images. And that shows that these decapping enzymes and argonaute are in the same location in the cell in these structures called Pbodies. So the point is simply this, that just like we saw with nuclear neighborhoods, Pbodies are present in the cytosol and are sites where mRNA is degraded. Now, what's interesting about this is that there's kind of a competition that determines what happens to the mRNA. And what I mean by that is this. We just saw Pbodies and we saw that they are sites of decapping and mRNA degradation. So let's say you've got this mRNA right here. Okay, one possibility is that mRNA could be translated, but that mRNA could also be moved over here to one of these Pbodies, okay, where it could be decapped and it could undergo degradation. So they're kind of like degradation factories, so to speak. But there are also these things in cells called stress granules, and stress granules are really interesting. Whenever translation of mRNA in a cell is blocked, for whatever reason, it could be that the cell is starved, and so those starvation conditions send a signal to the cell that it needs to be kind of quiescent or really not do anything right at the moment because it doesn't have enough food, enough resources. So translation just kind of ceases. It's put on hold. Some drugs can induce a block in translation, or there's some sort of genetic manipulations that can block translation. For whatever reason, if these mRNA cannot be translated, oftentimes they're moved over to these structures called stress granules. Now look at what stress granules contain. They contain mRNA. You can see the mRNA here, okay, with its cap on it. But they also contain translation initiation factors. They contain poly-A binding proteins, small ribosomal subunits. Now, if you've got translation initiation factors, poly-A binding proteins, and small ribosomal subunits, those are all things that are going to promote translation, right? Those are all things that are going to lead to mRNA stability and ultimately translation. So one way to think about stress granules is that stress granules are sites within the cytoplasm where mRNA become translation ready. So again, you've got these mRNA, maybe for whatever condition, for whatever reason rather, translation is halted. So these mRNA get stored in these stress granules. Now it turns out that mRNA that are in Pbodies can be degraded, but they don't have to be degraded. As this bottom line shows, they can also, under certain conditions, be moved over here to stress granules. And obviously, if they're moved over here to stress granules, then what's going to happen? Well, they're going to become translation ready and they could be translated. So this is really kind of cool. This is why you can describe this as a competition. It's like that mRNA could be translated, or there's a competition between movement to the Pbody or movement to the stress granule. And even when it's in the Pbody, there's a possibility that it will go to the stress granule. So the overall stability of an mRNA is determined by whether or not it's in one of these Pbodies or whether or not it's in one of these stress granules. What does all this mean? Well, it kind of seems like that what it means is that a cell is going to invest a large amount of energy and resources to produce a properly processed mRNA. Because it's invested energy, because it's invested time, because it's invested resources, it carefully controls the subsequent fate of that mRNA. In other words, once that mRNA is synthesized, fully processed, exported to the cytosol, it's as if the cell says, I got to be careful with this mRNA. It's a valuable commodity. I've invested all these resources, all this time, all this energy. I got to be careful what I do with it. Yes, it may be possible that it needs to be degraded because I don't need the protein anymore. But it may be possible once I put it over here in the Pbody that I want to move it out to a stress granule so that it can be translated. So the point is this, the issue or the determining factor on whether or not a protein is translated or not is complicated. In other words, it's not just a matter of whether the mRNA is going to bind and start translating it or not. It's whether it retains its cap, whether it retains its poly-A tail, whether it's in a stress granule, whether it's in a Pbody. All that stuff is going to matter. So you can see how important it is for the cell to control the fate of that mRNA. I think of it kind of like proteins. It's kind of like the cell invests all this energy into proteins to make these proteins. That's why if a protein is not properly folded, it's got mechanisms in place to try to ensure that it gets properly folded, rather than just saying, oh, well, that one didn't work. the same thing here. A cell's going to invest all this energy in making this mRNA. It wants to make sure it's used properly. Now what we want to do is kind of change gears a little bit and look at another kind of post-translational regulation. Post-transcriptional regulation. It turns out that there are 3 classes of non-coding RNAs that engage in a process called RNA interference. These 3 classes are called microRNA, small interfering RNAs, or pRNAs. We're going to talk about these 2, but it turns out that all 3 of these are engaged in this process called RNA interference. And so the idea is this: we've got a target RNA. This is our mRNA. Now, do we want to translate that or not? Now, we've already talked about some mechanisms that can be involved in determining whether or not it gets translated or degraded. So what we're talking about here is yet another mechanism, a really important mechanism as it turns out. So what's going to happen is that there's going to be this is kind of looking at this in general. There's going to be a double-stranded RNA. This is not an mRNA. This is one of these microRNAs or short interfering or pRNAs. This double-stranded RNA is going to be processed into a single-stranded RNA that's going to interfere with the target RNA. It's going to interfere with the target RNA by binding to it. And by binding to that target RNA, it's going to do 2 things that are going to inhibit its translation. One is it's going to lead to cleavage of that RNA, which of course we know will eventually lead to degradation, or it can lead to repression of translation and eventual degradation. Now, the third thing that it can do is a really, really interesting thing that really it's the same sort of mechanism, but it's not even, you can't even fairly call it post-transcriptional, because as we can see, what's going to happen here is it can cause the formation, these interfering RNA can cause the formation of heterochromatin. Now that's getting back to transcription, right? So these small interfering RNAs are involved in post-transcriptional regulation. That's what we're talking about. So I don't want this to become confusing, because they are involved in post-transcriptional regulation of protein expression, but they are also involved in transcriptional regulation. So let's look in a little bit more detail at how these work. The first thing we need to notice is that some of these, like the microRNA, are actually encoded by genes that are transcribed by RNA polymerase II. I always find that really fascinating, because the same RNA polymerase that's transcribing a gene that encodes protein is also transcribing a gene that encodes a microRNA that inhibits the formation of protein from the mRNA. So it's kind of like the RNA polymerase is making 2 things that are working against each other, kind of balancing each other out. So here's your mRNA, in this case shown in red. Here's your mRNA. Here's your microRNA. Now, we said that there were short interfering RNAs. There's microRNA and there's pRNA. I should say we're focusing right now on microRNAs. So these microRNAs are going to bind to mRNA, and they're going to have some effect that's going to block the production of a protein from that mRNA. They're going to do this in the context of a protein. So all this gray or silver, whatever you want to call it, this is a protein. You can see some alpha helices, some beta sheet, and some random coil there. So these microRNA, or this process of interfering with translation, was first discovered by 2 scientists named Mello and Fry. And they won the Nobel Prize for this work in the early 2000s. I don't remember exactly when it was. They discovered this in C. elegans, which is a nematode. It's a type of worm. And when they discovered it, people thought, wow, that's really cool. That's interesting. But it's probably just this novel sort of regulation that occurs in this these worms. Well, it turns out that the people that drew that conclusion were really wrong. It plays a big role in human cell biology as well. In fact, the human genome is now known to encode about 1,000 different microRNAs. And these microRNAs are responsible for regulating about 1/3 of all human protein coding genes. So this is not a trivial mechanism for regulating protein expression. It's a really important mechanism. So let's see more precisely or specifically how this is going to work. What's the sequence of events that goes on here? Well, here's what happens. In the nucleus, we're going to make this RNA. Notice it's got a cap on it. Notice it's got a poly A tail. It's double-stranded. In other words, it folds back on itself. So in other words, RNA are single-stranded, but because they're single-stranded, if the sequence is right, you can get base pairing, and that's exactly what you get. So these red lines here represent base pairing. So in the nucleus, this is going to get cropped, and this end is going to get removed, and what's left over is going to be exported out into the cytosol, where dicing occurs. And dicing is going to take off this loop on the end. Okay, and that's going to leave us with a short, double-stranded piece of RNA. It's only going to be about 23 nucleotides long. Now here comes Argonaut. Now remember, when we talked about Pbodies, we said Argonaut is going to be found in the same location as decapping enzymes, right? Well, take a look at what's happening here. The Argonaut is going to, and a bunch of other proteins, not just Argonaut, is going to bind to this double-stranded RNA right here. And one of these strands is going to be selected. And the strand that is selected is going to have a sequence that's the complement of the target RNA. So here's our target RNA right here. And so in this complex that includes Argonaut and the single-stranded short 23 nucleotide microRNA, you also now have the target RNA. And this is called an RNAi induced silencing complex. Okay, now once the target mRNA, the Argonaut and all the other associated proteins, and the single-stranded RNA are incorporated into this RISC complex, then what happens is the target RNA and the microRNA base pair with each other. Now, if base pairing is extensive, in other words, there aren't many mismatches, then what happens is you get slicing. And once you get slicing, by the way is clipping. You can see clipping right here on your target RNA. Once you get slicing, then the microRNA and the Argonaut proteins and other proteins are released, and this RNA can be degraded. But now remember, this is going to be taking place in one of these Pbodies. So one way to think about this is that microRNA are guides that bring the target mRNA into contact with exonucleases. So those exonucleases are going to be part of what you also would expect to find in the Pbodies. So in other words, one way to think about this is you've got these Pbodies that are sites of protein degradation, mRNA degradation, I'm sorry. The Argonaut is there because the Argonaut, along with the microRNA, have targeted or brought that target RNA over to the Pbody, and in the Pbody is where you have the stuff that's going to degrade the mRNA, including, as we saw, the decapping enzymes, which don't really degrade it, but we know that if we decap it, we're going to get degradation. you would expect to find exonucleases in those Pbodies, and then we can begin degrading that mRNA by chewing it up from both ends. So that's obviously a way to keep a protein from being synthesized by degrading that mRNA. Now, it turns out that in most mammals, that's not exactly what happens. Rather, in most mammals, the base pairing is less extensive. In other words, there are mismatches in the base pairing between the target RNA and the microRNA. And if there are base pairings, mismatches, then what happens is there's usually translational repression and eventual degradation of the RNA. But the first thing that happens is a repression of translation. But just as we see with the case of mRNA degradation, you're not going to get a protein. Whether you degrade the mRNA or you don't translate it, the end result is the same. That is, you don't get the protein. Now let's just look in a little more detail at what Argonaut is doing, since it plays such an important role here. It's got several domains, and you can see that it binds the microRNA. You've got one domain that's going to bind the 5' phosphate. And what it does is this Argonaut protein is thought to stretch out the microRNA gets kind of stretched out and gets positioned in such a way that it can easily form base pairs with the target RNA. And then you have this domain right here actually has the catalytic site where you can get slicing. Now, we said slicing doesn't occur in most cases in humans, but that doesn't occur unless you get the extensive base pairing, as we said. In most cases in humans, what's going to happen is this is just going to block translation. Now, there's one other thing that we need to talk about, kind of related to this whole thing, and that is that remember, we had microRNA, we had pRNA, and we had short interfering RNA. Well, let's now look at how the short interfering RNA work. They're going to work in a very similar way in the sense that they are short, double-stranded pieces of RNA. But they're a little bit different than the microRNAs. They can be incorporated with Argonaut and other proteins to form a RISC complex. If they're incorporated into a RISC complex, then we've already seen what can happen. We're going to get degradation of that mRNA. Or if you don't get perfect base pairing, repressional translation. But these short inhibitory RNAs can also associate with Argonaut and other proteins to give you not a RISC complex, but a RITS. And RITS, R-I-T-S, stands for RNAi induced transcriptional silencing. Okay, now this is what I was talking about a little bit earlier, because you ought to be thinking to yourself, well, I thought we were talking about post-transcriptional regulation of gene expression, but this looks like we're back to talking about transcription, and the fact is we are. But we're back to kind of talking about transcription because it's essentially the same mechanism, but the end result is a little bit different. Over here, it has to do with inhibiting translation or inducing degradation. In this case, what happens amazingly is that as RNA polymerase is transcribing a gene and making the RNA, this RITS complex, which contains now a single-stranded short inhibitory RNA, binds to this new nascent RNA. And when it does that, it recruits proteins that modify histones and direct the formation of heterochromatin. Now that's pretty amazing, because we know what happens in heterochromatin. If it's heterochromatin, then we're not going to get transcription, right? We can no longer get transcription because we have tight packing of the DNA. So in other words, look at what's happened here. if we have transcription of a gene, then that must mean that that DNA was decondensed, the gene was exposed so that the RNA polymerase could bind to the start site of transcription. But you're going to counteract that with this short interfering RNA that's going to be part of this Ritz complex that's going to immediately cause that to go back into heterochromatin where you don't get transcription. And you might ask yourself, well, what's the advantage of this? Why do we want to kind of have it both ways? Do we want to transcribe it or not? And again, remember, it's in the cell, it's usually not an all or nothing thing. The rate at which this particular gene is going to be transcribed in is going to be a function of what? It's going to be a function of a balance between those factors that are causing it to be transcribed and those factors that are causing it to be transcription to be blocked. So the bottom line here is that even though this is working at the transcriptional level, it's another way using these short inhibitory RNAs to affect how much protein you have. So that concludes this lecture, Lecture 10.